If the speakers for the next session are in the room, if you could come.
Good morning. We are ready to begin our next session. Our first speaker will be Simone Presque from State University of New York. I hope I pronounced that correctly. Her topic is water quality and phytoplankton communities of Bear Lake, New York.
So hello everyone, again I am Simone Lukasik and I will be presenting on water quality and phytoplankton communities of Bear Lake in New York. So the main purpose of this project was to develop a management plan. There's very little information known about Bear Lake because it's not collected and run monthly or routinely or even yearly. Last year in 2016 we started this project and another student went out from June to September to start collecting this data to build a profile of the lake. This summer we continued, starting in May and going until September to, again, build that profile to see exactly what the lake is doing and what it will do in the future. There are a few concerns from the community itself. People who live directly on the water have concerns about water quality itself, potential for harmful algal blooms, conservation, and fishing. And Bear Lake is a very popular fishing spot, and so they would like to keep that in the future. Also, what we tried to do is to gather foundational knowledge about the lake. As I mentioned before, there's not much known about it, even what type it is, whether it's mesotrophic, eutrophic, oligotrophic. We don't know much about the ecosystem, the phytoplankton community, or even the surrounding area and anthropogenic effects on the lake itself. So Bear Lake is a small headwater lake located in Chautauqua County, New York. It's really interesting because most of the shoreline is actually wetlands. So the wetlands cover about 70% of the shoreline. There are two main streams or tributaries that enter the lake, and there have been some conservation projects going on so far. Eurasian milfoil is a macrophyte that's an invasive species in this area and in other areas, and the community took it on themselves to try to control it, and they used weevils, which has been shown to be successful in other states. So the weevils basically suppress the plant growth, and it reduces its buoyancy, making it very hard to survive. Again, as I mentioned before, the lake type is not very well known. Right now, we're kind of borderline between mesotrophic and eutrophic. And as you can see in the chart below, the Secchi disk in 2016 was only about 2 meters, which would make it more eutrophic as water clarity is decreasing. And this year, it bumped up to about 3, which would make it more mesotrophic. And since it varied, it shows that we obviously need to do more work and continue this project to actually be able to identify it, and whether this is just an incident of 2 years or whether it will continue to vary. Also, another problem is that there's anoxia that's common in June, which I will talk about later. We think is mainly because of nutrient runoff coming from farmlands right on the tributaries. We also ran for chlorophyll A, and this is just an average comparison from 2016 and 2017. So in 2016, the chlorophyll A was about 24 micrograms per liter at a depth of 2 meters, and this year it was about 14. So the methods I used were to start GIS mapping, which I used using ArcMap. We did routine monthly sampling at 2 separate sites in the lake. As you can see in the rough image here, site 1 was in the deepest part of the lake, and site 2 was closer to the edge near one of the streams. At each site, well, at site 1, I should say, we went through 3 depths, 2 meters, 4 meters, and 6 meters. And at site 2, we did 2 meters and 4 meters. At each site and at each depth, we used soap wash bottles and acid wash bottles. The soap wash bottles were used to run for chlorophyll and phytoplankton, and the acid wash bottles were used only to run for nutrients as they're more sensitive. So the parameters we studied were nutrients, including nitrogen and phosphorus. Water quality was tested for basically using the YSI probe, and that includes probes that run for pH, chlorophyll, temperature, depth, and many more. The Secchi disk was used to study water clarity. Those are important, but today I'm going to be mainly focusing on chlorophyll, which was done using acetone extraction on the spectrophotometer, which was done following parameters set by the Environmental Protection Agency, and also phytoplankton counting. So to start, I mapped the watershed of Bear Lake. And the watershed information was from the TMDL given to us by the Department of Environmental Conservation. I generated multiple maps, including but not limited to this map here, which is land use, land cover. I generated one on geology to see the soils of the surrounding area, and a basic digital elevation model as well. So today I just wanted to touch on the preliminary map here of land use, land cover. As you can see, Bear Lake is right there. And the surrounding area is all in green here, is mostly your forests, which is good. You know, you get a lot of green, and that protects the lake. And right here is where we start to see some anthropogenic effects. The yellow is hay and pasture, and the orange is your cultivated crops. So hay and pasture will include your cows and your other creatures. And this is the actual like corn and wheat and grapes in the area. And that is what we think is causing some of the nutrient runoff problem into the lake, increasing our nutrient inloading. And right here, it's kind of hard to see here, but there's a little thin red strip. That little thin red strip shows moderate living conditions. So there's a moderate population of people there. That is mainly because right on the lake, there's campgrounds, and there's also the farmers who live there because their crops are there. Next, we studied bulk chlorophyll. And this is just a graph showing from each month has its own color and showing how the chlorophyll fared throughout the season. In May and September, which is your blue and your green lines, the max chlorophyll was seen at a depth of 2 meters, which is what I expected to see since more light is coming down for photosynthesis. But in June, July, and August, your other colors here, There's a chlorophyll max at 6 meters, which I found to be interesting as light cannot penetrate all the way down, so I thought that was kind of interesting. And over here you see like these really like sharp peaks, and I'll talk about this in a minute, but this is due to a species of phytoplankton called oscillatoria. So the phytoplankton community study was my main goal here, my main purpose. So this graph I generated was at an average of each of the 4-meter depths from site 1. So my most common species were Cryptomonas and Rhodomonas. Those are your two, those are your two blues at the bottom here, closest to the axis. Again, I didn't include 6 meters because this is just a 4-meter reading, but there is the oscillatoria blooms at 6 meters, which I wanted to make note of. This shows that there's very good diversity in the lake. We have our flagellates, which are the Cryptomonas and the Rhodomonas. We have the chrysophytes, cryptophytes, diatoms, which are your Asterianella formosa and your Fragillaria. We have chlorophyta, which is your Ancastrodesmus, and your cyanobacteria, which is your Anabaena, which we can see a big peak over here, which I'll touch on later as well. This also shows that it follows a traditional template for lake succession. We saw a very similar pattern in last year's data, including this peak in Anabaena in August. I just wanted to note also that I'm still counting for September data, and there will not be a comparison for today for chlorophyll. I wanted to include a couple of pictures of phytoplankton because they're my favorite. These are my 3 most interesting / most common. Over here in your far left, you have your Anabaena, and it is actually a nitrogen fixer. So what it does is when nutrient runs off into the lake, it sinks into the sediment, and during turnover, the nutrients come up again, and Anabaena just loves that. And that's where we get that big peak. If I go back, this is what is causing this big peak in August, is when that nutrients come up again and they eat it up and they just bloom. In the middle here, we have Asterianella formosa, which is a pennant diatom. This is definitely my favorite diatom because it looks like a star, and it is actually called, if you do a Google search on it, it'll say the star diatom. It tends to colonize and like glue together in this form all the time, and it can be bigger than this, it could be smaller than this, it varies. And then my last photo here is your oscillatoria, which, as I mentioned before, it's found at that 6-meter depth because it tends to regulate its buoyancy. So the 6 meter is like its happy place because it gets enough light coming down from the surface to do photosynthesis, but it also gets the nutrients coming up from the bottom of the lake. So you won't see it in like 2 and 4 meter depths, and then it'll spike in 6 and it'll go back down again. I wanted to do a quick comparison of phytoplankton compared to the chlorophyll. So over here are my same graphs. I wanted to make a couple notes. So there's a June spike in Asterianella formosa, that really big chunk of blue at the top here. And you can see if you look in your red zone here, there's a slight peak of chlorophyll compared to 2 meters, but it's not like relatively large. So I was not sure if it was exactly correlated with chlorophyll. It would be interesting to see it compared to nutrients coming in. And July had the most diversity. If you look, you can see all of your colors really well. And that was interesting because the chlorophyll really isn't that high for July. You know, it peaks at 6 meters, but at a 4 meter depth, it's pretty generic. So I was interested to see what exactly is causing that, if it's common or if it's just happening. So in conclusion, I wanted to focus on a couple parameters. So the watershed is mostly forested areas, which is good for the community as a whole and the habitat / ecosystem and ecology. There again is some agriculture, which could be causing the nutrient runoff and inloading into the lake. And that is coming from the farms that are cultivated and the pastures. As I mentioned before, the farms are located right on the tributary coming into the stream, and there are cows, and you know, they tend to use the water to excrete into and drink out of, which can also be causing more nutrient inloading into the lake than necessary. There are early and late season chlorophyll peaks closest to the surface, and there are middle seasons where chlorophyll peaking at the deepest depth. The dominant species were compared to the past data, and they measured up and related to things we have seen in the past, including the Anabaena spike in August after the fall turnover. Again, as I mentioned earlier, chlorophyll did not cause too much change in the phytoplankton community, It'll be interesting to see in future work if it does, and what exactly is causing the changes is unknown right now. It'll be interesting to see the nutrient data compared to the phytoplankton. In a big picture idea, like why do you care? Links between water quality and land use is what I wanted to mainly focus on. The nutrient loading into the lake is a big factor in your possible blooms that could happen, including the Anabena peak. Nitrogen is the main limiting nutrient in Bear Lake, at least in June, which is causing, again, that Anabena peak in August. Also, this lovely picture here is because Fredonia got a little flash flood this past weekend, I should say, and you can see over here there's the campgrounds that people tend to live on. Well, they're kind of non-existent right now. They're underwater, and this little boat launch is just a sign now, and this was all cement here to be able to pull in your boat, and it's just completely covered in water. And due to climate change and other storm events like this, it'll be interesting to see how the lake responds and what it will look like in the future. I just wanted to make a few acknowledgments to the Chautauqua County Soil and Water Conservation District, the Bear Lake Association, and the support for the grant. I thank them for their continued support in this project, and I would love to see what Bear Lake does in the future, as it's a beautiful little spot to fish and to just kayak. And thank you.
We do have time for a few questions.
Yes.
So in addition to measuring nutrient concentrations in the lake, did you also measure nutrient concentrations in the tributaries that were coming in and leaving the lake?
We did not, but it'll be interesting to see if somebody does in the future, to see exactly where it's coming from. I know I did one set of data on chlorophyll coming from the stream. I didn't study the nutrients, but the one stream is like where cows were directly like standing in. So it'll be interesting to see if, I didn't see too much of a peak in chlorophyll. I didn't include it in here because I only did it once, but it would be interesting to see if we could run nutrients on it to see. That's a good idea. Yes, oh, go ahead.
Question, you talked about anoxia.
Yes.
How long did it last and how extensive?
So I can't say exactly how long it lasted for sure, as it's hard to pinpoint when it starts and when it ends. Usually it's not too extensive. Usually when you have a bloom like that, it lasts for like a few weeks to a month. The lake itself doesn't bloom too much. We don't actually see covering on the lake, so it's hard to exactly say how long the anoxia lasts for, but I would love to talk to you after with my research advisor to answer that better for you.
Actually, there is anoxia that starts a little bit in June and it continues through August, but it's a weird temperature profile. I can explain a bit more as well. But yes, it starts in June.
So you did do a continuous oxygen profile then from through the season?
Yes.
So I noticed that you had that late season bloom of Anabena that followed the nutrient limitation as you described it.
Yes.
Is there any, and you also have a Silatoria, which is plankton.
Yes.
Is there any concern that the lake, these cyanobacteria may be producing cyanotoxins?
It's hard to say for sure, certain, as we don't test for the toxins in our lab directly. It is a concern of the community as it is a cyanobacteria. Bear Lake has a different type of structure. It doesn't really have a shoreline, so there's really nowhere for the bloom to take hold and actually cover the surface directly. We can try to bring in people who will actually test for the cyanotoxins coming from it. It doesn't seem to be causing a problem right now to the people and pets in the area, as the pets will go up and drink the water and they'll be fine. So it would be interesting to see in the future extensive study on whether it will continue to stay dormant or if it'll increase with changes in climate and changes in anthropogenic effects as well.
We have time for probably one more question. Okay, thank you.
Thank you.
You're our next speaker, I take it. Our next speaker is Heather Barrett, who'll be speaking on the effects of microplastic pollution on zooplankton. She's also from State University of New York, Fredonia.
Okay, hi, I'm Heather. So today I'm going to talk to you about the potential effects that microplastics could be having on freshwater zooplankton, in particular grazers. Just an overview, we're going to go around, we're going to go through the background information on Daphnia just so you understand a little bit about the organism and why we chose it, and just a little bit about microplastics. I'm sure a lot of people have been hearing about microplastics lately, and why we should care if they're interacting. So this is a paper done in 2013 by Dr. Mason. She's co-advising me. I just want to highlight part of it. We know that as the water flows through the Great Lakes, it's accumulating plastics all the way through. And the largest portion so far in this paper were found between Erie and Buffalo, which is right where SUNY Fredonia is. This is the amount of pellets that were found in this study that was done throughout several Great Lakes, but they were a huge portion of what was collected. And the largest amount of the pellets found were in this between 355 microns to just below 1 millimeter range. So zooplankton, we're concerned that this could be the likely entry point. We know fish and birds are eating microplastics, but we have been able to find much smaller pieces than we think they're able to just pick out of the water. These are a really important and key food resource for upper trophic levels. A lot of fish juveniles grow up eating these, and then even adults feed on zooplankton. So, grazers like these Daphnia control a lot of the bacteria levels as well as algae levels. So there's a concern that if their numbers are reduced because of influences on their feeding ability, that this could relate to property values and tourism decreases because of water clarity. When we're going into this, what we wanted to look at are mechanical and chemical effects. We know that there's degradation byproducts when plastics are affected to UV radiation, so there's a concern that this could affect their behavior with reproducing and feeding. And then with mechanical aspects, Daphnia are filter feeders, so there's the concern that this could inhibit their filtering. And there's well-documented cases where the increase in plastics are tied to a decrease in algal ingestion. This is one such, but this was done on North Atlantic copepods. The difference between copepods and cladocerans, which we're concerned with, is that copepods are very visual hunters. They can also be both herbivores and predatory species. This one focused on herbivores, but they use styrofoam. They went up to 30.6 micrometers. I say up to. A lot of zooplankton consume between 0 and 50 micrometers. And they documented it by looking at the algal ingestion rate, and they found that it decreased with increased plastic counts in the water. So in Lake Erie, we have cladocerans, rotifers, and copepods. Copepods, we have both the predatory and the herbivores. Cladocerans and rotifers are both exclusively herbivores. So what we're looking at for our project are these Daphnia. These are grazers. You could think of them as itty-bitty cows. They just sort of passively filter bacteria and algae out of the water. These are much less selective than copepods, although it has been documented that they can reject particles. But the concern is that with copepods that have a better vision, if they're eating microplastics, that these species, which are less selective, will also be consuming microplastics. Daphnia magna is the largest species of Daphnia. These are not, however, present in the Great Lakes. They are a really good model organism and easy to get through ordering systems. This is a graph I referenced a little bit earlier. So if you can focus on, so right there, that's the Daphnia magna. That's the species we're working with. They have been documented to eat over 5 microns, but it's uncommon. And then these Daphnia species, these are all present in the Great Lakes, and they have a very similar pattern. They generally prefer between 10 and 30 microns. Longer term effects, we are concerned that their reproduction could be affected by this. And this was done in 1996, but the clutch size is extremely affected by the food level intensity, whereas fish predation really controls the body length. So the thought is that if we see a decreased rate of algal ingestion, we should also see a decrease in clutch size. Our objectives going into this, we wanted to just get an understanding of how microplastics could be affecting zooplankton freshwater communities. A lot of research is done on marine communities, but around here we're more concerned about freshwater systems. And what we thought would happen was that zooplankton consumption of algae would decrease with increased plastic density due to either mechanical or chemical interferences. Most of the paperwork behind this supports this hypothesis. And then long term, we expected to see a decrease in their reproductive abilities. So what we did for our methods, we have this grazing wheel that is commonly referred to as the Ferris wheel. This makes sure that the daphnia, the algae, and the plastics all remain suspended. Because our zooplankton are very passive feeders, if the algae is not suspended, they can't really eat it. This only moves at 2 rotations per minute, so it's not whipping them around or anything, but it is enough to make sure that everything remains suspended. It can hold up to 40 bottles, so this would allow us to do much larger batches. However, it gets cranky when I put more than 15 on there. We've chosen the magic number 7 for zooplankton per bottle. 5 zooplankton really, they weren't eating enough algae for us to get a clear idea of what was happening with the ingestion rates. And when we got any higher than that, we were concerned about oxygen levels within the bottles. We started looking at 10 milligrams of plastic beads per bottle. This is, I just want to say this right now, this is much more than what is found in the lake, in even Lake Erie, where the highest concentrations are of microplastics, but a lot of our studies to date are observational, and we wanted to go with manipulative, so we just wanted to see if we could get a result. The algae we selected is Selenastrum. This is a single-celled algae. It's considered a really good food source for zooplankton. The previous study used polystyrene. We used polyethylene beads. These were collected down to 355 microns. This was just out of bathroom products that you could find on the shelf at the time. These were cleaned very thoroughly, so we didn't want any contamination with soap residue. The bottles held about 250 milliliters. This was a mix-up of algae and spring water that we make. And we started with one-day experiments, but in order to understand the reproductive issues that might come with this, it would have to be a bit longer, but what we've got so far are one-day studies. We were looking at optical density. This was taken at the beginning and the end, and we also looked at chlorophyll A levels, the same way Simona did, using the same method set out by the EPA. So what we found were the exact opposite of what we were looking for. These little balls right here are actually fat globules because they were consuming more algae with bigger beads in the system. So remember that they can only consume up to like a maximum of 50 micron sized bacteria and algae, and the beads we put in there were 355 microns at first. So well outside of their range of consumption. But we got much higher densities of these fat globules in the zooplankton exposed to these microbeads. There were a few present in the controls, but not nearly in the density or the frequency in those zooplankton. These are graphs we made, so it's the difference in the height overall is because it's very difficult to get the exact amount of algae in each batch as we go through different days. But there was this consistent difference in chlorophyll A amount. So if there's less chlorophyll A, it means that they're ingesting more algae. So this green one, this is the average. Those are the controls. Were on average higher amounts of chlorophyll, meaning there was more algae in the bottles at the end of the experiment. So where we'd like to go forward, we have ordered much smaller beads. They are between 10 and 27 microns. This is the perfect size range for mature Daphnia to consume. We would like to get into fluorescence microscopy so we could track the movement of the bead through the gut just to see if it gets stuck anywhere. We would like to see the reproductive effects. We are concerned about the oxygen levels in a bottle if we were to leave them on there for long enough to see reproductive issues. We looked at leachate. So far, we're not seeing any response, at least by the algae, to the leachate. Leachate takes a little longer to make because we're exposing it to UV radiation for a couple weeks at a time to make this spring water that has these chemicals in it. And right now, they're in a 24-hour light cycle. We would like to see what happens with light and dark cycle, just because zooplankton do behave differently at night. At least the Daphnia do. Just to round it up, we know plastics are a growing problem. It's really important that we look at the space of the food chain that's consuming algae and bacteria, because if the plastics are getting in at this level, we know that much smaller plastics can build up in the tissue of fish, and these are consumed by fish. So it's just one way that we're looking at it. And there's also a really important thing that we need to move away a little bit from observational studies and start getting into more manipulative studies to see if there's thresholds for what the ecosystems can handle. So I just want to thank, we are funded through the New York Sea Grant as well as an Illinois-Indiana Sea Grant. And I want to thank Dr. Harrington and everyone in the Mason Lab as well as everyone in the Wigdall-Perry Lab. And that's it.
We do have considerable time for questions, so
Yeah, sorry.
you're on the spot from here on.
I know. Okay.
What products are these plastics coming from? Is it bathroom product?
Yeah, so like face washes and body washes, a lot of these were sourced before they put the thing out about everyone not using microbeads. So it came from a variety of name brand products, but they were primarily face and body washes. Yeah. And we rinse them very thoroughly because the Daphnia are very sensitive to soap residue. So even when we're cleaning the bottles, we just rinse them very, very thoroughly with millicue water. We just don't want any type of biofilms building up. Yeah. Any more questions?
Other questions?
Yeah.
So I know some recent work that Dr. Mason did with the USGS in Great Lakes showed that it wasn't actually bees that were coming out of rivers, it was more fibers and filaments that were coming out of rivers.
Yeah.
So will that factor into your future research design?
There is an undergrad student, she couldn't be here today, she is focusing on fibers. She's having some difficulty teasing out the fibers because there's this myth that you have to put soap in your washing machine every time, so it took her about 5 times of nothing in her washing machine to get it to come out without soap in it. So we are working on the fiber issue. It's just that pellets were a very easy thing. We just wanted to see if there was an effect for their feeding rates and their reproduction. Go ahead.
Another follow-up question.
Okay.
So it is interesting that you had, if I understood it right, you had bees that were larger than what the Daphnia
Much
was
larger.
feeding on, right? Much larger.
Yeah.
But you saw that they had fat globules in the presence of the bees, but not in the absence, or a difference in the fat.
Right, so zooplankton that were exposed for 24 hours to beads, large, 355 micrometers or larger, were eating more algae. So they developed these fat globules, and they sort of, where they build up was, it wasn't in just one place, because some of them would get them in like their tail end where the chute is, and then some of them would even get them on their filtering apparatus. So that brings up a whole nother set of questions that if they are eating all this algae in the wild, these have never been documented, but the input of microplastics isn't really slowing down any. So this could potentially affect their ability to filter if it becomes more of a problem.
So were the globules also observed similarly in control without the bees?
No, it would be like maybe one, and that would be like a much larger zooplankton, because we did want to see how it would affect if we had different age ranges in there. So the first ones had different age ranges, and now we're using they're all between a week and 2 weeks old. And if a control did have one in it, it was a much older zooplankton.
Other questions?
Yeah, that's fine.
Okay, we have 5 minutes before the next presentation, so I'm going to allow Grove City then to load their material onto the network. Our next speaker is Caroline Fideli from Grove City College, and she'll be speaking on the response of brine shrimp to light in disturbance. Caroline?
Hello. Today I'll be talking to you about some research I've been working on at Grove City College with my collaborators Elizabeth Schwab, Ken Kato, and Eric Anderson. The purpose of this research is to determine if our method of data acquisition and processing is effective in the study of small aquatic organisms. We're actually working with a type of zooplankton, brine shrimp, and we use the known behavior of brine shrimp called phototaxis, which is the act of swimming towards light, to test our turbulence tank and particle tracking techniques. Here's a picture of the turbulence tank. So, why do we care about brine shrimp? We don't really. We're using them to better understand oyster larvae settlement and dispersal, specifically by exploring physical factors like light and turbulence, which might govern those behaviors. The work is expected to contribute valuable information to ecologists, hatcheries, and marine resource managers. Oysters are extremely significant to the ocean's ecosystem as they clarify and filter water, and they're also an important food species. What we want to know is, are the factors of light and turbulence, do they cause them to swim down and settle? Do they affect them at all? So here is some previous work on oyster larvae collected at Woods Hole Oceanographic Institution. And what this graph is saying, well, there's two different data sets. This is the first one with one batch of oyster larvae. And it's depicting the vertical swimming velocity here on the y-axis. So upward is, or positive is swimming upward and negative is swimming downward. And then we have the turbulence level here measured in the energy dissipation rate. Each color just represents a different trial, and we see that the oyster larvae are kind of hovering in the water when there's no turbulence. And then for higher levels of turbulence, they tend to be swimming upward, according to this top graph. Down here, this next batch of oyster larvae that were tested, again seem to be hovering around zero velocity at a zero turbulence level, and then are kind of all over the place. So we want to better understand, does turbulence affect, does it cue them to swim downward and settle? So our methods and materials, we want to know if our experimentation method will yield valid results if we test it on oyster larvae. So we're using brine shrimp, and these are the materials. Jet stirred turbulence tank, which we place the brine shrimp in water, and then also an LED flashlight to create the light that we know they'll swim towards. And then the camera and the infrared laser, basically the infrared laser will shoot a two-dimensional sheet of light into the turbulence tank, illuminating all the particles, brine shrimp and the tiny particles in the fluid, which we use to track, I'll explain later on, which we use to measure the fluid flow velocity. And then the camera is taking strobe images, about 50 to 100 images per second, at the same rate the laser is flashing. So basically we get like a strobe image sequence, or basically a video to track each brine shrimp's movement. So here we have a video of the brine shrimp in the turbulence tank in still water. There's no turbulence in that video. And here's one of the image sequences of the brine shrimp. And as you see, they're kind of coming in and out of focus. And this will be important later on. But the background particles that you see here are actually little plastic micron beads that we use to track the velocity. So, yeah, it's just short-term experimentation. They do actually ingest the plastic. But here is some of our data. This graph right here is showing a trajectory of one brine shrimp's movement in an image sequence. So we have the vertical and horizontal velocity of its overall velocity relative to the camera. And then right here, we used a different software it's the same shrimp right here, same surrounding fluid. But we want to know the velocity that the surrounding fluid is going at. So this graph the little arrows are indicating the direction, and the color is indicating the speed of the surrounding fluid. So what we're trying to find is the behavioral velocity of the brine shrimp. Is it actually swimming upward or downward? Not is it being carried upward with the current, but trying to swim downward. So when we have the surrounding fluid velocity, we created what we call an annulus graph, which indicates what the fluid is doing in the immediately surrounding area. And that's what we call the mean fluid velocity in the vertical direction. So down here, we have an equation to calculate the behavioral velocity of the particle, of the brine shrimp, where we take this net velocity of the shrimp in the fluid and subtract this mean velocity of the surrounding fluid from it. So basically, and here, so the brine shrimp is going at 1.99 millimeters per second. The surrounding fluid was going at 1.1 millimeters per second. So that gave us a net result of 8.9 millimeters per second in the vertical direction. And basically what this is saying is for example, if the brine shrimp was going up at 2 millimeters per second, going upward overall, and then we calculated that the fluid was going upward at 1 millimeter per second, then the brine shrimp must actually be swimming upward at 1 millimeter per second because it's going faster than the fluid. So our results, the effective light and turbulence. Each point on this graph that in the red indicates a set of data taken with light, and each point in blue indicates a set of data taken without light. The vertical axis indicates the behavioral velocity that we just calculated. Again, above 0 is swimming upward, below 0 is swimming downward. And then the horizontal axis indicates the turbulence level that we were testing it at. So here we see that both with and without light, the brine shrimp are swimming downward. And then next, at a low turbulence level, they're hovering around 0 without light and still swimming downward with light. And then later on, they appear to be hovering around 0 with light, swimming upward without light. And then lastly, at a higher turbulence level, they appear to be swimming up in both cases. And this, at our low turbulence level here, this was the only point that was statistically significant between the two with and without light sets that were taken, because you don't see an overlap in our 95% confidence interval here. And we would have done the same if we had more time for each of the other data sets, but basically each of them extend a 95% confidence interval about the same length as this. So what we got from this graph is that we can't really determine if the light is affecting their behavior or not, because as you see, in both cases, they're swimming upward at a high turbulence level. But what we can determine is that there's quite a significant difference between this, their motion in still water versus their motion in a high turbulence level. So what we concluded from this experiment was that we can't conclude the effect of phototaxis. The data, we can't conclude if they're swimming towards the light. But they are swimming upward in higher turbulence and downward in lower turbulence, as we saw in the previous graph. But when we were testing this, when we put the brine shrimp in the turbulence tank with the light, we could actually see them swimming towards the light. So we know that they do exhibit phototaxis. But why don't we see this in our results? A few explanations could be that we just didn't collect enough data. So since each of those plots on the graph was the mean of the data, it could be that the outliers or random brine shrimp that were going in the wrong direction canceled out the effect of the others or altered that mean. Another explanation is that when we're shooting this laser and taking the image, the reason that the brine shrimp were coming in and out of focus is because they're going in front of and behind this laser sheet. So it's shooting a sheet of light into the turbulence tank and taking an image. But when they're not directly in that sheet of light, they're going to be out of focus, and that could affect the calculations that we got. And then also possibly our data, we might have not gotten the initial response of the brine shrimp when the data was taken. And then also another explanation, our turbulence tank has 8 actuators that produce a uniform isotropic turbulence, at least they're supposed to. And if there's a slight variation between the top and bottom actuators, it could produce either an upward or a downward turbulent flow. And then the brine shrimp might exhibit different phototaxis behavior. So we don't know if when there's turbulence, that might affect their behavior towards light. Future research, we'd probably conduct this experiment again, taking a lot more data, and start the data acquisition right when the light goes on. Because as we saw in the oyster larvae graph earlier, it was hundreds or thousands of oyster larvae being trapped. Our data was between 10 and 100 brine shrimp being trapped. So we definitely need a lot more data. But lastly, I would like to thank the Grove City College Jewel Moore and Mackenzie Fund, awarded to Eric Anderson for our flow visualization system, and also Evan Lundberg in Dr. Anshin's lab for helping with data processing. Thank you.
We do have time for questions.
Yes.
So you mentioned that it might be that there's differential between the jets that would cause an upward flow instead of just like everyone mixing, right?
Yeah.
So I guess I'm just interested, do the brine shrimp just tend to kind of go with the flow, right? When there is a flow, do they tend to track the flow? That's why they change from swimming down to swimming up when there's turbulence. You know, I guess I'm just trying to understand like the behavior of it. Like they were swimming down when it was still, and then they completely switched directions when there was turbulence. So is it more that they're tracking the flow and they tend to just sort of swim where they're being pushed, or?
Well, our graph was specifically the behavioral velocity, so that's why we subtracted out the turbulent.
Right, so that's what I'm saying, but their behavior completely changed direction.
Yeah, yeah. So we're not sure if, well, we did it. to some extent, they do tend to just kind of drift to the bottom when they're just in still water and there's no light. They kind of just drift to the bottom. So the first set of data at zero turbulence is what we expected. Swimming upward with higher turbulence, that could, again, I said the jet or the turbulence tank with actuators might need to be recalibrated and made sure that there's no upward current flow. But that's something new that we weren't really expecting to see swimming upward.
Yeah, it's really interesting.
Yeah.
Just curious.
Yeah, and we did expect them to have more of the swimming towards the light.
Right.
But we don't know if does turbulence affect phototaxis? Like if there's an upward stream, do they kind of stop swimming because they realize they're going towards the light? And so they might just kind of be hovering and riding the current. While in still water, they actually have to swim to get to the light. So, yeah. Yes?
So kind of building on that, what if you kept exactly the same jet setup, but you switched the side of the light? Wouldn't that, I mean, because then you can account for the fact that there might be differences in turbulence top to bottom. So
Yeah.
you'd be testing phototaxis counts.
Yeah. Well, to account for
That's a thing.
the difference in we'd probably just want to recalibrate our turbulence tank and make sure that the levels at the top and bottom are the same. But that would be interesting in seeing. Our data didn't really show anything for phototaxis. It was more they exhibit different behavior for low turbulence versus high turbulence. But that would be interesting to see. If we did switch the light, would we see anything there? Would we get any different results? But, yeah.
Random thought, no one can about your field.
Other questions? We still have 5 minutes before the next speaker, so.
I don't know. So I don't have any idea about this, but you said it's an LED light?
Yes.
Are there different effects from phototaxis depending on the wavelengths of light that are emitted? Like would a different kind of light elicit response? I don't know anything about that. But
Yeah. And that would actually be really interesting to see if they exhibit different behavior with different frequencies of light.
I'm just wondering if you use an incandescent versus an LED,
Yeah.
or, you know what I mean? If there would be different wavelengths
Yeah.
of light that we emitted from those, but if it's a certain wavelength that they can, they can only
Yeah, they can't
sense
see
a
the
certain-
wavelength as
if
well.
their receptors only sense certain wavelengths, then you're not giving them that would explain why you don't have phototaxis.
Yeah, that's very possible. We did use an infrared laser because that's not visible light and we didn't want it to affect their behavior. But we didn't really look into what wavelengths of light they really respond to best. I don't think. Yes.
How well can rhinos see? Because like some organisms as small really only can detect light direct.
Yeah.
But sometimes there's like shapes and
Yeah. I'm not entirely sure. They definitely do exhibit phototaxis, like just sitting in the tank when we had just like shining a flashlight on one side of the tank, we'd see them all gather and collect. And it's like some of them would start right away and like just swim right towards it. Some of them would actually just kind of stay. The majority of them would swim towards the light though. But I'm not really sure what, again, like the frequency of light could definitely have an effect in how well they see it. But,
Other questions? I think this is the mark of a good research project. It opens up more questions and more research opportunities. We do have about 2 minutes, so in order to keep everything on schedule, the next presentation will be in about 2 minutes. So. getting ready for the next presentation. The next presentation will be presented by Abigail Knoll from Grove City College. The title of her presentation is The Balance of Fluid Forces in Fish White Propulsion During Deceleration, Acceleration, and Constant Speed Swimming. Abigail?
Thank you for that introduction. I just wanted to clarify something in my title, the fish-like propulsion. In this experiment, we didn't actually test live fish. We were testing a flapping plastic strip, like you can see up here at the top, that was mimicking fish undulatory motion and propulsion at different swimming cases that I will explain later. So I want to go into why it is important to study a flapping piece of plastic when we're really interested in fish. So this can have biological significance in energy budgets. So to understand the energy that a fish needs to swim, it's helpful to understand how much drag is on the fish, how much drag the fish is overcoming at different speeds of swimming. And this can also be extended to populations in ecology. So as an energy budget can explain one fish, it can also be expanded to fisheries, migration, and therefore how much biomass a fish will need to consume in order to have enough energy to swim. This can also have technological significance in biomimetic vehicles. So we could copy the undulatory motion of a fish to create more flexible oceanographic research vehicles and perhaps make them more efficient. That is the question we are seeking to answer through this research. So why study forces in fish swimming? So the first reason is solving Gray's paradox. Now James Gray in a 1936 paper studied dolphin swimming and estimated the drag power on a swimming dolphin and compared that to the available power in the dolphin's muscle mass. So what he found was that the available muscle power wasn't enough to actually overcome the drag on the dolphin at high speed swimming. So this posed a problem for the dolphin that came to be known as Gray's paradox. And this also was related to work that was done on bees that compared the lift that the bee was producing with its wings to the amount of weight on the bee and led to this saying that was kind of a colloquial saying of Gray's paradox, fish can't swim and bees can't fly. Now we know that's not true, so how do we reconcile Gray's calculations? One of the ways is by saying Gray's drag power estimates were wrong. That would decrease the drag power and the dolphin will be able to overcome the drag at high speeds again. A second way would be by saying Gray's muscle power estimates were wrong, which would then cause the force balance to be, again, in the muscle power's favor. And then finally, another way of reconciling Gray's paradox would be to say something about the dolphin swimming, the undulatory motion that it's using to propel itself, is causing this magical reduction in drag that we don't know about, and that might be another way that the drag could be reduced, and therefore the dolphin could be efficient again at those high speeds. However, in a paper by M.J. Lighthill, Lighthill estimated that the friction drag should actually be 3 to 5 times higher in undulation than on still bodies. And the friction drag being the interaction of the fluid with the object's surface, and that causing drag on the object. So if the friction drag is 3 to 5 times higher in the undulation than on still bodies, that causes our force balance again to move in the direction of drag. So our work is seeking to check Lighthill's theory. Is the drag on an undulating object actually higher than the still body? And we're going to do this by calculating the friction drag on an undulating panel like I mentioned before, and you can see over here on the right. And doing this, we will also determine the full balance of forces on this undulating object. The breakdown of the friction drag, which again is the interaction of the fluid with the surface, and the pressure drag, which is the drag due to the changes in pressure around the body and the flow. So in order to study the friction drag, we look at the boundary layer region near the surface of our undulating plastic strip. I want you to look up here at these particles in this flow field that have green circles around them. As you can see, they're all in about the same vertical place in the field, and this is the plastic strip that was undulating in the previous slides. Now, as the flow moves forward, you can see that the particles near the surface didn't move very much, and the particles farther away moved a lot further. And this is caused by the interaction of the friction of the foil and the fluid, and it leads to a boundary layer profile, which is what we call this shape of the flow that is caused from the motion and the interaction with the foil. Now, what this tells us, the slope of this linear region here, you can see that it's linear up to a point and then it kind of levels off back up to the free stream velocity. And the slope of this region is used in drag calculations. So by taking flow fields around this undulating plastic foil, we can calculate a lot of these slopes and then use that information to calculate the friction drag on the panel, on the foil. So for our methods and materials, this is what the plastic strip looked like. It was 19 centimeters long, and the top view, just like you could see in the videos of the foil moving back and forth. We swam this strip under a number of swimming cases, the first being non-flapping. So the plastic strip was connected to a biomimetic robot that would heave the plastic strip back and forth for the swimming cases, a frequency of 1, and for the non-swimming case, the frequency of 0. So the non-flapping foil was swum at 0.1 meters per second, 0.135 meters per second, and 0.17 meters per second. And by the speed, we mean the speed of the water in the flume. The plastic strip was tethered. So when I mean accelerating, constant speed, and decelerating, which were swum at 0.1 meters per second, 0.135 meters per second, and 0.17 meters per second, respectively, I don't mean that the foil was actually moving forward in the flow because it was tethered, but I want to explain this with an analogy. So if you imagine a swimmer in a swimmer treadmill, one of these endless pools, if you have the flow moving across the swimmer and the swimmer is moving his arms at a constant frequency, that swimmer is going to produce a certain amount of thrust as if he were moving in a pool that doesn't have flow moving past him. So he's staying in place. This is what we call our constant speed swimming case. So when the thrust that the swimmer is producing is matching the speed of the flow. Now, if you imagine that the swimmer continues to move his arms at the same frequency, producing the same amount of thrust, and we increase the speed of the flow, the swimmer's going to fall back in the flow. And that's what we called our decelerating case. And if you decrease the speed of the flow, the swimmer would move forward. Now, if you tether the swimmer, like we tethered our plastic strip to a harness, and the speed of the flow is increased, the swimmer's going to want to move back, but he's tethered, so there's going to be a force on that harness that we measured on our plastic strip using a transducer. So now we know how much drag is on the strip and how much thrust the strip is producing at the lower speed cases. So this is a sample of what the data would have looked like. And this flapping strip, as you can see, we took data zoomed in here at different positions. along the length of the foil. And by measuring the boundary layer profiles, as I mentioned earlier, we were able to determine the drag over the length of the foil. This is a sample of what one of the boundary layers looked like. We generated thousands of these from, again, different areas on the foil. This was taken halfway along the length of the foil, so the red square that was in the middle. And we used these profiles to, again, calculate the slope of this linear region, which told us information about the drag. And you can see that on the y-axis, this is the distance from the foil. This is on the order of a centimeter. We're taking videos that are just a centimeter high, and the boundary layer region that we're measuring is on the order of millimeters. So it's a very small region that we had to zoom in really far on the foil to be able to measure. So here were our results. For the friction drag, as you can see, the not flapping cases, so the still plastic strip, as you increase the speed of the swimming, the drag increased, which makes sense. We would expect that to happen. However, at the accelerating, constant speed, and decelerating cases, the drag on the panel was higher in all of these cases than the respective non-flapping case. Now this is interesting because this tells us that the friction drag on the undulating object is actually higher than on the still object. So this seems to prove light hill correct. And you can see in the accelerating case, this is more in depth on the accelerating case. If you imagine this to be the front of the strip and this to be the back of the strip and this the length along the foil, and the y-axis being the shear stress, which is a rating of the friction, as the foil, as you move along the length of the foil for the flapping case, you can see this increase in drag, and for the still foil, the drag is always lower than the flapping foil. So this again supports what we just found, that the non-flapping cases shows an increase in friction drag in all areas of the foil, but especially the back. Now I want to talk about the back of the foil. So as you heave this plastic strip back and forth, you can imagine that the end of the foil will have a higher amplitude, which basically is more flapping. So we even see more drag in more flapping, which again supports the idea that undulation is increasing the friction drag on the object. So this is the constant speed case, and you can see a really similar effect. We have one point that is a little different, but for the most part, the drag is greater on the flapping case than on the still case, and you see this increase towards the end of the foil. And then finally, the decelerating case, again, a really similar trend, maybe one point that looks different, but the drag is higher for flapping than it is for non-flapping. So here is data for the full force balance that I mentioned earlier. Total drag is made up of friction drag and pressure force, like I mentioned earlier. And as you can see in this plot, the total of the friction drag plus the friction drag plus the pressure force is equal to the total force on the object. And this is just interesting information. This is the first time that we know of that this information has been able to be found, because it's so hard to separate out the pressure force from the friction force. Usually you can calculate the total force on an object in flow just by the transducer measurements that we mentioned earlier, but separating out the friction and the pressure requires a lot of information, like we mentioned earlier with the boundary layer, and so this is kind of the first information of its kind, and we're working through what that means right now. So in conclusion, we did measure an increase in the friction drag on an undulating object, and this does support Lighthill's theory. So it agrees with Lighthill and argues that Gray's paradox must be solved on the basis of power measurements and not a magical drag reduction that would be caused by undulation. So this also agrees with work by my advisor, Dr. Eric Anderson, on swimming fish. He found a very similar effect with swimming fish and measured the friction drag on swimming fish and found that it was also greater in undulation. So what this means for biomimetic vehicles, if we want to increase the efficiency of biomimetic vehicles by making them swim like fish, it might not be the best way to do it. Because if there's more drag on an undulating object than a still object, there probably are better ways of increasing the efficiency of a vehicle like that. However, there may be other reasons why fish swimming would be a good model for a biomimetic vehicle, like maneuverability, the ability to get into tight spaces like underwater caves and that type of thing. I would like to acknowledge the Jewel Moore and Mackenzie Fund Award that funded our research, and the Harvard Museum of Comparative Zoology, where I had the internship that allowed me to take this data in the summer of 2015. Thank you.
Do you have time for one or two questions? Anybody?
This is really interesting. I'm just processing. But I'm thinking about the nature of the surface from like a fish tail versus a dolphin skin, and then you're using plastic to look at the mechanics. But what do you think about different types of materials and how that might influence that drag? You know, the issue of drag at the surface.
Yeah. So a lot has been done after Grace research into this maybe magical drag reduction. A lot of work has been done on like shark skin, swimming suits, and that sort of thing that might, again, decrease the friction drag. And the surface does play into it a bit, but we're seeing that the undulation actually changes the friction drag even more than those effects of the surface, because the boundary layer, like I was mentioning earlier, the surface, so a lot of that is actually caused by the friction of the fluid on the fluid and less with the surface itself. Because when you get down close to the surface, the particles are not moving a lot, no matter kind of what the surface looks like. And now the surface can change that. There's a lot of work that's done into kind of like slippery coatings on fish, so we haven't really looked into that much, but it definitely is an interesting question that maybe in the future we can look into. Are there any other questions?
No questions? Okay, if not, our final speaker has a dubious honor of going just before lunch. So, but our final speaker for this session is T.L. Gridley, and he'll be discussing the review of flume speed ramping as a method for correcting tail beat kinetics during fish swimming.
Thank you for the introduction. First off, I'd like to thank my collaborators. Abby Knoll did some work on this field, as well as Carl Dannenberg, who was presenting here last year, and then our advisor, Eric Anderson. So this work is part of a larger work looking at resonant frequencies in fish swimming. Similar experiments to what Abby was just talking about with the flapping panels, finding certain sweet spots or like ideal ways in which these panels can move. So the question that arose from finding this resonance in the swimming plastic strips was, do these same sweet spots appear in fish motion. So the importance of that research is that it can lead to a better understanding of fish behavior and ecology. If the length and the stiffness of the fish affects the efficiency of its motion, that can give a better understanding to why they behave as they do. So how we collected this data is, I believe, the same flume that Abby had her panel in. As seen here, it's this treadmill or wind tunnel for the fish to swim in. The water is pushed through at certain speeds. It's capable of speeds from 0 to 2 meters per second. And the setup that we use to capture the 3D position of this tail is a stereoscopic setup with 2 cameras at different angles such that we can get that 3-dimensional position of the fish tail. And from that position, over time, that provides us with the frequency and the amplitude. That is how fast it's swinging its tail and how wide of strokes the tail is flapping at. To get the data needed for the fish resonance project, we need to run the fish at hundreds of flume speeds to get the full picture of where it has ideal zones or likewise, but I'm not going to be talking too much about that, just rather the supporting data collection for that. So because we need those hundreds of speeds collected, the time it takes can be very long. Our original method is the one seen here on the left, where we would run the fish at set speeds for a while, change the speed, run it at that speed, change it again, and this specifically has the higher speeds slowly ramping up one at a time with some rest periods, taking data both at the higher speeds and at the rest periods, running the fish for a while, then taking the data so that the fish has time to get into that and the flume has time to get up to speed. The ramping method that I'm going to be talking about and that we've experimented with is much smaller increments of the speed. So if you notice here, we have the flume speed in meters per second and time in minutes. And looking at the scale of the Y, you can see in the ramping, we're just in that narrow upper area as these top lines are at. And then looking at the x-axis with the time, seeing that ramping, we're going many more different discrete increments of speed at a much shorter period of time. So this would allow us to get a lot more of those speeds in much less time. Again, just supporting all of the research relating to the fish swimming kinematics that cares about running at different speeds. These are the 2 fish that the data in this presentation is explicitly talking about. There are 2 striped bass, they're going to be called bass 4 and bass 5. There's some basic dimensions if you care, pretty similar fish. Yeah. So here is the data capturing method. Again, it has the 2 different cameras, the one at the angle and the one straight in, to get that 3-dimensional position of the fish. And you can see the point that the software is following as the fish is moving. And that point, again, is captured 3-dimensional data. And here you can see, just plotted out, the cross flume position, or rather the direction that the tail's wagging over the time. And this is positioned inside of that box, the flume box. So looking at this data that we collect, we can, again, find the frequency. We use the power spectrum to find the frequency, and then by finding the peaks and the troughs, we can get the amplitude of how far that tail is wagging. And to get the overall speed at which the fish is swimming at any point in time, we take the velocity of the flume, which was found with some other work, and add the velocity of the fish in the flume, again, since we have that position, looking at the average position to figure out if it's moving in the flume, to count for if it's moving in the flume as well as the flume speed itself. Taking all that data, we arrive here. This is tail beat frequency versus swimming speed for bass number 4. On your y-axis, we have our tail beat frequency in hertz, or rather, how many beats per second the tail swimming at, and the swimming speed in the flume, how fast the fish is swimming. Our blue squares here is the non-ramped data for bass number 4. Again, the blue graph that you saw earlier on with the different plateaus over a larger period of time, and then the orange is the ramped data, or the stepwise graph over the shorter period of time. And they both follow these, they both have these nice linear fits to them, which makes sense that if the fish is swimming faster, that it will be moving its tail faster. And then we have the similar data for bass number 5, or bass 5. Again, same axes, blue squares being the non-ramped, orange squares being the ramped. Same linear fits. The lines are very similar, although there is a slight difference in the slopes, which we'll be talking about. But by looking at them and seeing that they are very similar, we already get an idea, okay, this ramping method is giving us similar results to the non-ramping, so it may be viable for us to use this. Taking a closer look at the slopes, we arrive here. Again, we have the bass 4, ramped case for the bass 4, bass 5, ramped case for bass 5, and then the slope, or the magnitude of the slope on this axis here. And pay attention to the values here because this is somewhat zoomed in, so they somewhat exaggerated difference in the slope. We have also the 95% confidence intervals as the black lines around these. And there is, from this graph, you can see a statistically significant difference between the slopes for the ramped and the non-ramped cases. And in both cases, the ramped data is a lower slope than the non-ramped data. So this would, looking at this makes us wonder, okay, is this actually, are we getting different data from the ramped versus the non-ramped? So, as said just then, slope of frequency versus swimming speed in ramped cases is lower. Why is that possibly? Because of how quickly we were taking the data, it may be that the flume was not up to the speed we thought it was when we were taking the data, and as such, the fish was actually needing to swim slower than we recorded on the data, and as such, that would skew the data and make it so that fish doesn't need to swim as far, and it makes sense then that their tail is moving less. That would be one source of error where it was methodological error, and we will look in the future to correct that. So future work will be taking detailed data of the flume during ramping to find out how quickly the flume gets up to that speed that we expect it to get up to, and then once we find that response time for how fast it gets up to speed, we'll ramp accordingly, giving enough time for the flume to get up to speed before we start taking data on the tailfish position. Also, by taking the data of how fast it comes up to speed, we may be able to correct the current data by seeing how fast the flume was actually moving when that data was taken, rather than the assumption that we went off of that it was up to its speed. So, once we do these corrections, ramping is again looking like it may be a viable option to get data faster, since the slopes are similar and we know of this methodological error, and we'll be looking into if we can fix it and if those slopes line up closer. So altogether, it seems that ramping would be a useful and workable way to collect fish tail kinematics faster for future studies with fish resonance or other things looking for that type of data. In conclusion, I'd like to thank 3 of the people working back at Grove City with us, helping to process data, as well as funding from the Sweezy Fund to Grove City in support of Eric Anderson's lab at the college. Thank you for having us today.
We do have time for questions. Any questions?
So just as a follow-up on your kind of issue of like a secondary measurement of ramp speed, I guess. So the flume itself doesn't have an independent measure of water velocity in it. It's only controlled by like jet speed or I don't know anything about these.
So let's just bring back for visual. The flume is powered by an impeller or something pushing the water, and the measurement of what we set the thing is at is a motor speed in hertz. And it's increments of 0.1, so we'll run it at 10 hertz, 10.1 hertz, etc. We don't have any, I don't know of any other speed measurement in there that we have directly at certain speeds, but what we do is measure with putting little plastic beads into and finding the particles and using software to find the velocity of the water speed at certain fan speeds, but we don't have something running at the same time as the fish is in there currently.
I have a question. You said there was a significant difference based on what statistical test did you use on that?
That's based off of the 95% confidence intervals and seeing that they don't overlap.
Oh, okay. Other questions? Okay, if not, enjoy lunch.